What if I told you a human organ can be emptied out like a building? No tenants, no furniture, no photos on the wall. And yet, the entire architecture remains. And then, here's the wild part, scientists try to move new living cells back in, like a carefully planned biological rehousing program. This isn't science fiction, it's a real pathway in regenerative medicine. Strip an organ down to its extracellular matrix framework, then reseed it with living cells. Today, I'm taking you deep into the rabbit hole. But don't worry, I'm leaving breadcrumbs so nobody gets lost. The trail map. Promise and structure. Here's the trail. 5 checkpoints. The blueprint, what the framework actually is. The demolition, how cells are removed without destroying the organ. The plumbing problem, why blood vessels decide everything. The repopulation, reseeding cells and training them to behave. The reality check, what works today, what's experimental, and why ethics matters. All right, let's begin. The blueprint, what's left after cells are gone. Inside your organs is an invisible city plan called the extracellular matrix, the ECM. It's a complex scaffold of proteins and structures that tells cells where to attach, what shape to take, which direction to grow, and what neighborhood they're in. Think of it like this: a crowd of people versus the stadium they're in. Cells are the crowd. The ECM is the stadium. The tunnels, the seats, the exits, the entire logic of the place. And here's the kicker. The ECM doesn't just hold shape. It preserves the organ's internal architecture, including vascular pathways, the routes blood would normally travel. So the dream becomes, if we keep the stadium, can we invite new players and restart the game? Checkpoint number 2, the demolition. Decellularization. Decellularization is basically organ exorcism. We remove the living cells because cells carry most of the immune ID badges that trigger transplant rejection while trying to preserve the ECM structure underneath. So how do scientists actually do this? One major breakthrough was perfusion decellularization. Instead of scrubbing the organ from the outside, scientists push detergents and solutions through the organ's own blood vessels, like running a cleaning cycle through a building's plumbing. In a landmark experiment, researchers decellularized rat hearts by perfusing detergents through the coronary circulation. The result was a preserved, perfusible vascular architecture and the overall geometry of the heart still intact. It's like running a dishwasher cycle, except the dishwasher is a heart and the plates are your hopes and dreams. Checkpoint number 3, the plumbing problem. Why blood vessels decide everything. Now we hit the boss level, vascularization. You can build the most beautiful organ scaffold on Earth, but if oxygen and nutrients can't reach every corner, cells die. And organs aren't thin like paper, they're thick, layered, hungry. That's why scientists love decellularized whole organs, because the vascular road network is already there, at least in structure. But structure is not function. A blood vessel isn't just a tube, it's a living surface lined by endothelial cells, carefully designed to prevent clotting and regulate flow. Without that living lining, the body looks at the vessel and basically says, "Congratulations, here's a clot." Checkpoint number 4, the repopulation, recellularization. Now comes the thrilling and humbling part. You've got an ECM ghost organ. Now you try to bring it back to life. Recellularization means repopulating that scaffold with living cells. Endothelial cells for the vessels, parenchymal cells, the organ's main workers like heart muscle, liver cells, or lung epithelium, supportive cells, fibroblasts, pericytes, and immune modulating cells depending on the organ. And you don't just sprinkle cells like salt on fries. Scientists often perfuse cells through the vascular system, inject them into specific tissue regions, then culture the organ in a bioreactor, a life support chamber that provides flow, oxygenation, pressure patterns, sometimes even mechanical stretch or motion. Basically, welcome to your new house. Also, you'll be paying rent in ATP. The "wait, this actually worked" milestones. Milestone A, the bioartificial heart. In early proof-of-concept work, decellularized hearts were reseeded with cells and maintained in perfusion bioreactors, demonstrating early organ-level engineering function. Not ready for human transplant, but enough to prove scaffold plus reseeding can produce functional tissue behavior. Milestone B, the bioartificial lung in animals. Here's the cinematic moment. A decellularized lung scaffold was reseeded with epithelial and endothelial cells, then transplanted into animals, showing gas exchange in vivo for a limited time. That's not a finished therapy, that's a prototype, like the Wright brothers flying for 12 seconds and humanity realizing, okay, so the sky is possible. The Dark Valley. Reality check. Why this is hard. Now, truth time. Whole organ decellularization and recellularization is real science, but it's brutally hard. First, cell sourcing. You need massive numbers of the right cell types, ideally patient-specific. Stem cell-derived cells are promising, but consistent maturation and safety are still major challenges. Second, endothelialization is everything. If the vessel lining isn't right, thrombosis and inflammation become immediate threats. Third, organs are ecosystems. A liver isn't just hepatocytes. A lung isn't just alveoli. You're rebuilding multi-cell choreography. Fourth, quality control and regulation. You're not making a gadget, you're making a living therapy. That means sterility, residual DNA control, immunogenicity testing, reproducibility, and regulatory oversight at an extraordinary level. In other words, it's less like printing a document and more like rebooting a civilization. The ethical sidebar. Science doesn't happen in a vacuum. Some tissue-engineered organ stories entered public imagination too fast. A well-known example, a tissue-engineered airway transplant in 2008 used a decellularized donor trachea plus cells from the recipient. Later, parts of the broader trachea engineering saga became intensely controversial, sparking criticism around evidence, oversight, and patient risk. So we hold two truths at once. The platform is real science, and translation to humans demands extraordinary caution. What's real today? Where this already helps patients. Here's the key distinction. Whole replacement organs, heart, lung, kidney, are still largely in research and preclinical translation, but decellularized ECM materials, patches, grafts, and repair scaffolds are much more established in clinical and translational medicine today. Why? Because they're smaller, simpler, and easier to vascularize safely. So this technology isn't all or nothing. Sometimes the future arrives as a patch before it arrives as a full organ. The future roadmap. If this pathway succeeds, it likely happens through convergence, better stem cell pipelines, patient-derived cells that mature reliably, smarter bioreactors that mimic real organ physics, flow, pressure, stretch, better endothelial lining strategies to keep vessels open and stable, hybrid approaches, decellularized scaffolds, plus organoids, plus advanced biomaterials. And along the way, we'll see stepping stone wins, engineered airway segments, liver assist grafts, vascularized tissue modules, personalized disease models for drug testing.
The future may not begin with, "Here's your brand new lab-grown heart." It may begin with, "Here's a living graft that buys you 5 more years, then 10, then a full replacement." Somewhere right now, someone is waiting for an organ donor. And somewhere else, a researcher is holding a scaffold, an organ's silent architecture, trying to persuade life to return. This is the strange, beautiful frontier. We're learning to separate the blueprint from the body, and then asking if we can rebuild the body from the blueprint, which is either the most hopeful science on Earth or the most elaborate way to say, "Nature, please let us borrow your instructions." If you want part 2, I'll go deeper into the detergents and decellularization standards, how bioreactors actually teach cells, why lungs
are harder than hearts in some ways, and what patient-specific organs would truly require. Like, subscribe, and comment which organ should we explore next: kidney, heart, liver, or lung? Drop your question below, and I'll see you in the next story.